D, E, and K. Liver produces most proteins needed by the body. Liver prevents shortages of nutrients by storing vitamins, minerals, and sugar.
Okay. Is it clear from the written functions? He has narrated, self-explanatory. Thank you very much, Doctor. Have a seat. So we will start with the most diverse and versatile cell located in the liver called hepatocyte, right? So it has to do a lot many function, 100 many functions the single cell is supposed to do, right? And let me select some other picture to go through about the grocery. If you want to have a look on it, you know it is located or occupying your right hypochondrium, isn't it? And extending definitely into epigastric and other region as well, right? So if you see, it's being covered on its outer side by a thin connective tissue capsule, glycan capsule. And what it's gonna do? It will enter or send septa or trabeculae inside to divide the major organ into small lobes and lobules, right? Okay. So let's see what large lobes it has. It has got, if I put a line just to demarcate, that is the left lobe and that is the right lobe, right? In addition to these two large lobes, it has got two small lobes, which are quadrate and quadrate, respectively, right? And definitely later on we will study the inner structure. But here you can see another important thing that two vessels are entering towards the liver through hilum. Hilum is a base for entry-exit, you know. Okay, we can see that portal vein is entering that blue color and hepatic artery. So let's discuss that portal vein carries how much amount of blood? Can anyone guess? Yeah, 72 to 75, right? And hepatic artery will take rest of 25%. And what is going to happen? That portal vein is selecting or carrying blood from your, can you name the organs please? Yeah, GIT, spleen, pancreas, intestine, very good, right? And what is going to happen? That all nutrient rich but deoxygenated, right? Blood is going into your liver. Now, from where comes the well oxygenated blood? From 25% through this hepatic artery. But when they enter into your liver, before reaching towards the hepatocyte, that versatile cell that is resting inside, the blood mixes up. So we can say that the individual hepatocyte, the cell, is not getting fully oxygenated blood in its life because it's being mixed before. ठीक हो गया? Is it clear? Still doing its function. कोई गिला शिकवा नहीं, हमें ये नहीं मिला, हमें ये नहीं मिला. There is a mixed blood, still doing its function very well. Another important thing, so that you must know that gallbladder is present here, producing a knot along the inferior border and having the duct system. You will study along with it. Okay. And you can see the connectivity as well. That there lies the inferior vena cava. So we should know that how blood supply is supposed to be moved inside. Let's select some other good picture. Okay, for learning purpose, we study liver into three, you would say, small lobules. Number one is the classical liver lobule or classical hepatic lobule, right? One way of studying the liver. There is another type of lobule which is called as, let me just first name them, is this triangular, the portal lobule. Can you see it? Okay. And if I divide or draw another one like this is now lozenge shape or diamond shape. This is the hepatic acinus. So we can study liver in three different ways. because it is important for anatomy, for physiology, for pathology, and for functional and for surgeons as well, because they have to dissect, we have to study different functions. Okay. As we have seen that what function the liver has to do, it must be having some organelles inside to carry out all these functions. So let's first review those to get some good picture related to that. Very beautiful picture, schematic picture you would say of a cell. And if we imagine that it is a hepatocyte, what we see, it has got a basal surface or basal domain. You understand the word domain? Yes or no? What is domain?
Function, structure.
Not function, not structure, it is the side or surface. So that is the basal domain. And we can expect this is the apical domain towards the lumen. Right? Okay. What we see? We see the nucleus, I mean the nucleolus, and all around you observe the rough endoplasmic reticulum. Why it is called rough? Because yeah, the ribosomes start along with them. And what they have to do? Yeah, protein secreting and binding factories, and they give intense basophilic stain to that basal region of the hepatocyte, right? If you see that hepatocyte, and that hepatocyte depicts the apical region, or collectively the eosinophilic stain. Why? Because of the extensively present mitochondria and smooth endoplasmic reticulum as well. And of course, we have got some glycogen granules over there. And when you give a stain, they get dissolved or wash away. Vacuolated appearance could be there. And what special about its nucleus? It could be showing uh, diploid number or polyploid, means 6 to 8 times more number of inside. You could say the combination of the chromatin material which appears as a scattered heterochromatin clusters. This is a hallmark or particular feature of your hepatocyte. Sometimes you observe binucleated cell as well, right? These are the features. Why I'm putting stress on it. Sometimes a question can come that draw, or what do you know about the ultrastructure of the hepatocyte? So the structures you see under the electron microscope and enumerate their function. When you know that there is smooth endoplasmic reticulum, what it has to do, it takes part in the detoxification. And you know the bilirubin, which is insoluble, should be converted into soluble one, so that it must be excreted from your body. And what else your this hepatocyte has to do? It behaves as endocrine, right? And as well as exocrine. So we can say that this liver, a single organ, is endocrinal and exocrinal. Exocrinal by producing bile, because it uses the duct system. You know now that exocrine needs ducts, right? And what about the endocrine? It has to produce various plasma proteins, ferritin, albumins, so many other things, fibrinogen, so that the body should function properly wherever there is needed. And we see that it is also there, the liver, to store various vitamins, especially vitamin A, or we can collectively say ADEK, the fat soluble. But what about that D? Is it stored inside? Yeah, you observe it there, but it is not stored, rather being supplied to your skeletal adipose tissue, right? Then whenever it's needed, being provided to your body. So we should know each and every function. There is much more detail you will learn in your text manuals. Okay. Okay. Now we see that how we have to further study after the ultrastructure, the organelles and the function you already enumerated. Right. So let's can see a cut section of the liver, and what you are observing, the classical hepatic lobule or classical liver lobule, hexagonal in shape, means 6 sides, which I have drawn on the whiteboard as well. What you see in the center, there lies a central vein or inlet venule, you can call it. And what another thing you observe, if this is the slide of mammal, you get a well-marked connective tissue around these lobules that demarcates one lobule or separated from the other. But if this is the slide of a human, there is very sparse or very less amount of connective tissue that you hardly observe in between them, and you see that one lobule is seems to be connected with the other. You get my point? This is just like a floor of made up of so many tiles, right? And if you observe it from far away, you can see that there is a one larger block, isn't it? But when you get closer and if you see the markings around every tile, you see that connective tissue and you observe the separate that lobule. Get my point? Okay, thank you. Now, on the corners, what you see, there are three structures in this area called as portal space or portal area. Carrying a branch of hepatic artery, could be a branch of portal vein, and of course the bile duct. So these three structures, they are forming the portal triad. Triad means three things, right? On the 3 to 6 corners. And important thing, now in this hexagonal structure, what we observe? We see a single plate of hepatocytes running through and through. Let me clear it on the blackboard. If that was the central vein, right? And we observe these plates of hepatocyte, single brick of wall, anastomosing. You have seen a wall made up of single brick of wall. Usually what happens, आपने देखा होता है कई दफा दो ईंटों की चुनाई से आप दीवार की तामीर देखते हैं, but here only single plate is being formed by the adjacent hepatocytes. And what is the shape? They have got polygonal shape. खुद ये hexagonal था 6 sides, but individual cell is polygonal. Means more than 6 could be the sides. We never know. Is it clear? Now, you can see them running from all sides like sun rays. If I draw like this, can you see? Okay. Now, intervening area, in between them, we observe these dash I have made. What this is area for sinusoids. And what is sinusoid? Can anyone help me to tell? Capillaries, fenestrated sinusoidal capillaries. This continuous endothelial. You can see the gap in between, isn't it? So having the endothelial cells, if I complete, and another variety of cell they carry are the macrophages, hence here called Kupffer cells. What they are supposed to do? They have to eat up the effeated or aged old blood cells, and in the meanwhile they have to collect the inside. If there is an iron, it's being stored as a ferritin complex, right? And overall that hepatocyte is going to store the lipid as well, and we observe glycogen inside, and there could be the other processes going, sometimes glyconeogenesis or glycogenolysis, so and so. When we fast, or जिसका आप देखते हो रमजान में, that stored glycogen will be the later on source of your glucose, that becomes very healthy to provide energy to your body, right? Okay. And we were just discussing the sinusoid. Sinusoid क्या है? ऐसी खून की पतली नालियाँ in between these plates, right? But you are seeing another thing, that there is a gap, still gap, or there is a space in between the hepatocyte and the sinusoid. So what it could be? That is the space of Deci or peri-sinusoidal space. इसको कैसे हम understand कर सकते हैं? If this is a sinusoid, and if I make these are two hepatocyte plates like this, can anyone see? All of you? Now, the space around this is peri-sinusoidal space or space of Deci. Is it clear? ये plates हैं, दरमियान में साइनोसॉइड था, लेकिन इसके इतराफ you get this space, this place and this one. Is it clear? And what it is made up of, or what clinically it is giving any significance? It is the real place where plasma exchange occurs. The blood is going and the hepatocytes are supposed to produce various substances. So real exchange plasma or substance exchange takes place in this space of Deci. Hepatocytes have got the microvilli that projects in this space of Deci, and from here you get the interaction of blood. So this is the real place of plasma or material exchange. Okay, what lies here? Here we expect a special type of cell, the stellar shaped cells, and we call them as ito cells. They are meant to store vitamin A retinol की form में. ठीक हो गया? अच्छा, what else is there? Here we expect the reticular fibers and the protein type collagen 3. Okay, last time the question came, university question came about the classical liver lobule, that in cirrhosis what is going to happen? Cirrhosis is due to the underlying fibrosis, and what histological changes we expect? That collagen type 3, which is normally present, is being now replaced by the collagen type 1 and 2, and they are supposed to be a reason of fibrosis and leading to cirrhosis of the liver, right? So that's why we sometimes mention, or we must read the clinical or medical applications related to that, because they just explain that space carrying the ito cells. Then you should know that this is the space of Deci. And what it is present around the sinusoid 1 definition, or it lies in between the sinusoid and hepatocyte. And it's the same thing, and you have to write what it carries inside, what type of collagen normally present, and what happens in the cirrhosis. Is it clear now? Okay, that was all about the arrangement of it. We observe a thing that usually in our life, that the center has got the main things, and that central area, central city, central country, or the authoritative person spreads things towards the periphery. देखते हैं शहर में चीज है और सेंटर में है आपके, और वो पेरिफ्री में जाती है. ऐसे होते हैं, ऐसी सप्लाई होती है ना. यू हैव गॉट लाइट सोर्स विद द वापडा. जब लाइट जाती है तो वापडा की तो नहीं जाती, ये शायद एट द एंड जाएगी. पेरिफ्री जहाँ सप्लाई है वहां चली जाएगी. इट हैपेंस. ओके, बट as we have seen that there was the portal triad carrying the blood vessels, but the rule of Allah, He has directed the flow of blood from periphery towards center. A beautiful approach, right? From periphery towards center, because the blood is now entering into the sinusoids finally, and it will drain into this inlet venule or central vein. And if this is a superior aspect, if I turn it like this, and you see the central vein is going to be draining to sublobar or lobular veins, and hence the hepatic veins, and that hepatic veins finally approach towards the inferior vena cava. And can anyone tell where that inferior vena cava is going to be entered or opened?
Right.
Yeah, into your heart. So all the food, halal, haram, what we eat, is collected through the portal vein, entering or being scanned through the liver and through inferior vena cava, again back towards your heart or in your blood. Is it clear? Okay. But there is another interesting thing. We have to talk. We just mentioned the thing, the blood supply, the cells, the details. But what about the flow of bile? The bile is being produced no doubt inside. The factory is again the hepatocyte. But where it is? There are small bile canaliculi. The hepatocyte is going to produce bile, and they are coming this bile canaliculi, right? And these bile canaliculi, they will coalesce and they will become larger to form the canal of herring. This bile canaliculi doesn't have any epithelium. सिर्फ क्या है कि इसके दोनों तरफ हेपेटोसाइट ही इसकी दीवार बना रहे हैं. जैसे किसी तरफ के दरवाजे खुद से ना हो, साइड वाली चीजें उसको बाउंड्री दे रहे हो. दो हेपेटोसाइट उस बारीक बाइल कैनालिकुलाई का बाउंड्री बना रही है. नो सेपरेट epithelium. Again a question, again an MCQ could be. MCQ rather, right? Okay. And now when they are coalescing, they are joining to form larger duct system, or I mentioned a canal of Hering or bilateral bile ductule, sometimes same name. They are now lined with a simple cuboidal epithelium. That epithelial cells now called as cholangiocytes. Or you mention among them, there lies the oval cells. And what they do? They do the function of regeneration. You have seen, you must seen that in liver, sometimes liver transplant is being advised. This is the liver, and you resect a portion, or you take it from a donor, you match and put it on a recipient. What happens? That the donor portion again regenerates very well, and the patient receiving that portion of it can regenerate, so that full lobes are ready to work again. So what are the engineers, scientists that are making are these oval cells or cholangiocytes in the canal of Hering, and the hepatocytes that are performing the function of progenitor cell. Is it clear? That can again be asked that what cells are doing the function of this regeneration. So far is clear. So we any question, doctor? Is it clear? Okay, we'll take question at the end. Okay, because it's much important topic, easy to identify the slide, but for learning you have to concentrate a little bit more. We talked about the three ways to study this liver. So let me show you a schematic diagram taken from your Janpura. So you can see these hexagonal classical liver lobules, right? And you see that they are taking blood from periphery towards center, isn't it? Now this green color triangular structure being marked as portal lobule. Classical liver lobule address करी थी endocrine function, is it clear? Portal lobule exocrine function address करेगी. Why? अब आप देख रहे हो the central structure here is the branches of bile duct, hepatic artery and portal vein. और इसके edges पे तीन side पे क्या है? Central vein आ गई है. If you study like this, now the flow of bile is reverse or opposite to that of blood flow. Blood flow is from periphery to center, and bile flow is from center to periphery. So the purpose of taking this triangular area to study the exocrinal function. Is it clear? Now we have to study the third most, the hepatic this acinus, which is the diamond shape or lozenge shape. Okay. Here what you observe or what we study the supply of blood, and in addition to it we study the oxygen concentration or oxygen gradient, right? So for this, kindly consider it because it's very important. If you observe the periphery of it, which is being marked in this pink color or eosinophilic stain, kindly concentrate for more because it's very much important. That is called zonation. So zone 1 cells, which occupy, if I draw from whole side this pink color, is the cells or hepatocytes of zone 1, which are near the periphery. We could say the periportal hepatocytes, right? And another variety being marked with this blue called as zone 3. And if I complete like this, they are around the central vein. So they are the hepatocyte or cells of zone 3, right? So definitely intervening leftover is zone 2. Okay. Now we say that so many things are being produced and reach that hepatocyte. So that cells of the zone 1 are first to receive blood supply, first to receive nutrients, first to receive the even toxic effects of various drugs or various food or nutrients. Like, it's clear that first to receive. So whenever there is necrosis, ischemia or no blood supply or anything, what is going to happen?
the cells of zone 3 will first to die. Get my point?
Yes.
कि इरिगेशन सारी पेरीफ्री से आ रही थी, ठीक है, फुल सप्लाई पेरीफ्री की तरफ है. जैसे ही पीछे से सप्लाई कट ऑफ हुई, पेरीफ्री अभी मुतासर नहीं होगी, सेंटर से पहले सेल फारिग होना शुरू हो जाएंगे. यू गेट इट? द सेल्स ऑफ द जोन 3 आर फर्स्ट टू डाई. Okay, and here they are the cells to face the insult of fat accumulation as well. Fat necrosis accumulation could be here. Okay, if we observe that blood supply is revived, so the first cell which will regenerate will of zone 1. अभी इनकी जैसे लाइट गई ही नहीं थी, जा रही थी, फटाफट आ गई. दे विल फर्स्ट बी इरिगेटेड. सो फर्स्ट टू रीजनरेट, लास्ट टू रीजनरेट. राइट नाउ फर्स्ट टू डाई, लास्ट टू डाई. इफ सो, सिंपल इज द रूल कि जिसके पास जो अथॉरिटी है, जो चीज है उसके पास है. जैसे ही दूसरी जगह की स्विच ऑफ होती है तो अकॉर्डिंगली द सेल्स विल बी अफेक्टेड. और डेफिनेटली द इंटरमीडिएट जोन विल डेफिनेटली शेयर बोथ थिंग्स. ये ना तो फर्स्ट की लिस्ट में आ रहे हैं, ना ये लास्ट की लिस्ट में आ रहे हैं. इंटरमीडिएट. सो व्हेनेवर देयर इज अ इस्केमिक नेक्रोसिस, myocardial infarction or so and so, and blood supply is impaired, so liver could be one of the place, and cirrhosis too, and that happens. So very important that through this hepatic acinus we learn about zones, right? Is it clear? And through this portal lobule we study the exocrine function. Is it clear? And through this we study the endocrine function. We never know which part will come in the next exam if they are still focusing the liver. Okay, one or two more diagrams to make the concept a little bit more clear. Okay, that was a relevant figure. Let me show you. Kindly concentrate on some. That was another topic. That is a gallbladder, a pear shape. You can see just like a color is even being. Place to store bile. Bile was formed in the liver but stored in the gallbladder. Whenever we eat the fatty thing, what happens? The signals are reaching towards the vascular externa of this gallbladder. It starts contracting and pours its all content through its duct called as cystic duct towards that duodenum. But how? Now see that the left and right hepatic ducts coming out from the liver, they join to form common hepatic duct. Left and right will form common hepatic. That common hepatic is joining with the cystic. C is joining with the C. Is it clear? Okay. Now what they are going to form? They form CBD, common bile duct, and that is opening into the second part of the duodenum in the major duodenal papilla. The food we are taking and is reaching through esophagus, through stomach, and through duodenum is being or helped out by the secretion of this bile that will help to digest. Isn't it? Okay. Any other diagram? Let me show you. Yes. Now we will see. Kindly concentrate for a few minutes. These are the hepatocytes. Can you see? And intervening, these are smaller bile canaliculi. Can you see? छोटी छोटी नालियां, सड़कें, गलियां कह लें. They are coalescing to form the bile canal of Hering. No epithelium here individually, but bile canal of Hering are addressing simple cuboidal epithelium. You get it? Having cholangiocyte, oval cell, the regenerative cell inside. Okay. Now they are coalescing to form the bile ductules. And finally, they are reaching towards the bile duct on that portal triad. और वहां से आप देख रहे थे लिवर से लेफ्ट एंड राइट हेपेटिक डक्ट निकली थी. गॉल ब्लैडर की सिस्टिक डक्ट उसके कॉमन हेपेटिक डक्ट से मिली थी. सीबीडी बनी थी, कॉमन बाइल डक्ट, जो सेकेंड पार्ट ऑफ ड्यूडेनम में खुली थी. सो आपको ये डक्टल सिस्टम याद होना चाहिए. दैट विल हेल्प टू explain the exocrinal function कि बाइल ने इन सड़कों से, इन गलियों से मोटरवे तक चढ़ना है और डेडेनम में पहुंचना है. Is it clear? Okay, thank you. I think that is more. Now we have to study the slides. Okay, slides are a little bit. Kindly focus that. Okay, what slide is in front of you? Can you guess?
Liver.
Yeah, liver. What helps you to identify at one glance?
Lobules.
Yeah, classical liver lobules. We are on 4x, and you can see the central vein and these darker stain running single-plated structure are hepatocytes. And the white space is the sinusoid, and you can see the dotted cells could be the endothelial or Kupffer cells or macrophages. Okay. Now, what you see on the edges are the connective tissue well marked. And if I focus that triangular area, that would be the portal space of portal canal addressing the portal triad. Let me focus it on. Here, this is the place. We can see a larger dilated. That would be the vein. There is another could be the arteriole artery structure, and that could be the bile duct. Let me move towards the high part. These are the hepatocytes, right? And this is the intervening connective tissue, which is present in other mammals other than human. So this is the central vein. Now you can see the radiating plates of hepatocytes, intervening sinusoids having the Kupffer cells, and this is the connective tissue. Let me focus the portal triad area. Any corner you can select to see the. Yeah, this can be a beautiful picture to show the artery, the vein, and the bile duct. This more prominent bile duct. This could be artery, and this is the dilated vein and intervening connective tissue. Okay, doctor. Another important thing that this is the row which is peripheral row of hepatocytes or zone 1. So in between it and the connective tissue, there lies a space of mole. It is being said that lymph is being produced here, right? The lymph is present or being produced in between the peripheral hepatocytes and the connective tissue. And this particular space is called as space of mole, right? Okay. And these cells are busy in protein synthesis. Okay, zone 1 के cell थे. So that was about the slide of liver. Now we have to see the slide of gallbladder, an easy structure, but we have to learn the points of identification. We have seen the gross picture along with the liver. Okay. Now what you see from mucosa. This mucosa is thrown into various foldings, and you can observe rather the diverticula in between. These are the mucosal folds, not villi, right? And lying with the simple columnar epithelium. And few sparse microvilli could be there, right? And this area is the lamina propria.
Yes.
No muscularis mucosae. Get it? No submucosa. Direct, these irregular clumps are muscularis externa. Along with this irregular bundles, the smooth muscles, we have got intervening elastic fibers in addition to collagen fibers. Why this is random arrangement? I've told you when we were studying the stomach, the most random arrangement, there is another hollow organ, the gallbladder, having again a bulk of smooth muscle randomly. It gives contraction from all sides because the bile is being thrown through the cystic duct finally into your duodenum. Is it clear? And Allah has placed so many elastic fibers to help out or to accommodate that bile content. Is it clear? And finally, we reach up to the cirrhosa or adventitia. When it is present on the inferior aspect of the liver, being adventitia on its aspect, and below can be lying with the cirrhosa. And you already know the difference between the cirrhosa and the adventitia. So what these are? Can anyone guess what these rounded Lumen structure.
No.
Can you guess
No.
what these could be? This one, this one.
Nerve to no.
नाव का ल्यूमन इस तरह दिखता है.
नहीं.
What else says lumen?
Artery.
Yeah, the blood vessels could be artery, could be vein, having the blood cells inside. Isn't it? Okay. The gallbladder does not have any glands except if its neck region is being cut. Okay. So that was all for today. Thank you.